Have you ever wondered how scientists were able to conclude that DNA is the genetic material? That DNA is the one that carries the blueprint of life. Now we will discuss the sequence of experiments that led them to conclude that yes, it is DNA, not RNA, not proteins that carry genetic information. The first of these experiments was carried out in 1928 by a guy named Frederick Griffith. Frederick Griffith used two strains of pneumococcus. One strain is virulent, represented here as the S strain, and the other strain is non-virulent, represented as R strain. So what he did was he prepared 4 cultures of pneumococcus. First culture is made up of the S strain that is alive. The second culture is made up of the R strain that is also alive. The third culture is made up of S strain heated, thereby killing the bacteria. And the last culture is made up of a mixture of the living R strain and the virulent or the deadly S strain, which was also heat treated. So you have living R strain and dead S strain in the fourth culture. So he induced these cultures to 4 samples of mice. The results are that in the first culture, the mouse dies because of the virulent S strain. For the second culture, the mouse is alive because this is the non-virulent R strain. For the third culture, the heat-treated S strain, he observed that the mouse remained alive. So it means that because of the heat treatment, all of your S strain bacteria are dead. So they cannot cause death in the mouse. However, in the last culture wherein we have the living R strain and dead or heat treated S strain, the mouse died. And the only thing that could explain this is that there is a living S strain in this culture. So there must be something in the dead S strain that was transferred to the living R strain, causing it to be transformed into the virulent S strain. That is why Frederick Griffith coined the term transforming principle. It is the thing that is being transferred from the dead S strain to the living R strain, causing the living R strain to become a living S strain and causing disease and death in the mouse. In the 1940s, 3 scientists performed another experiment that also added evidence that DNA is the transforming principle observed by Frederick Griffith. So these three guys were Avery, MacLeod, and McCarthy. They also used the same bacteria used by Frederick Griffith. So they have the S strain of the pneumococcus. So this is the virulent strain. So they prepared heat treated S strain. So these bacteria are already dead due to the heat. And then they removed the lipids and sugars from the culture. And what you have left there are your proteins, your RNA, and your DNA. So they separated this solution into three. In the first solution, they added proteinases, and these enzymes destroy your protein. So there are no protein molecules in the first solution. In the second solution, they added RNases. These are enzymes that destroy your RNA. So you don't have RNA in the second solution. And in the third solution, they added DNases, enzymes that destroy DNA. So you don't have DNA molecules here. And then they added R cells or the R strain, the non-virulent strain of the pneumococcus, into the culture. And what they observed was in the first solution where you only have RNA and DNA and no protein, S cells appeared. In the second solution where you only have protein and DNA, so there's no RNA, S cells also appeared. But in the third solution, wherein you have protein and RNA but no DNA, S cells appeared. And this is a very compelling evidence that DNA is the transforming principle observed by Frederick Griffith. So their conclusion was that DNA is the one carrying genetic information that transforms the non-virulent R strain to the virulent S strain. In addition to the experiments and the observations made by Griffith and Avery, MacLeod, and McCarthy, in the 1950s, two more scientists were able to add evidence that DNA is the molecule carrying genetic information. So we have Hershey and Chase. Hershey and Chase worked with bacteriophages. So these are viruses that invade bacteria. So they used radio labeling. They used radioactive phosphorus to label the DNA molecules because your DNA molecules contain phosphorus, right, in the phosphate sugar backbone. So they used radioactive phosphorus to label DNA, and they used radioactive sulfur to label proteins because we have sulfur in proteins but not in DNA. So what they did was they have the, the radio labeled the virus or the bacteriophage with the proteins radio labeled. They allowed this to infect bacteria, Afterwards, they centrifuged the culture, so the bacteria will be in the precipitate while the bacteriophage will be in the supernatant. And then they tested for radioactivity in both the supernatant and the precipitate. So because the bacteria are relatively heavier than the bacteriophages, the bacteria will settle as precipitate. So when they tested for radioactivity, they noticed that there is radioactivity in the supernatant but not in the pellet or the precipitate, which means that when they labeled the proteins, they didn't transfer through the bacteriophage, did not transfer any radio labeled proteins into the bacteria. So the second setup was they have the bacteriophage whose DNA was radio labeled. So again, they allowed this bacteriophage to infect bacteria. And then after infection, they grew this in a culture. So you have the culture grown with the bacteriophage having DNA radio labeled. And then they centrifuged it and found that there is radioactivity in the pellet, but not in the supernatant. So it means that the bacteriophage transferred something into the bacteria, and this something would be DNA. Because in this second setup, they labeled the DNA with radioactive phosphorus. Since they were able to detect the radioactivity in the pellet, it means that DNA was transferred from the bacteriophage to the bacteria. And this is again another solid evidence that DNA is the is the molecule carrying genetic information.